My name is Madam Jennifer from Educare TZ. Welcome my lovely Form 1 students in our lovely subject biology, and we are starting our new topic known as viruses, kingdom Monera and kingdom Protoctista. And our topic comprises 3 subtopics, which is virus, kingdom Monera and kingdom Protoctista. My lovely Form 1 students, in this topic we will be aware on different types of viruses, on how they are transmitted, those diseases which are caused by the viruses, how they are transmitted, how can be cured, and how can be prevented. So you are mostly welcome in this topic because you will be aware on different diseases, their causes, their transmission, and how to prevent them, and become a good teachers in our community. You are welcome. Now, by starting with the first subtopic, which is viruses. The virus is an extremely small fragment of nucleic acid, which is DNA and RNA, surrounded by protein code. So these are just a small fragment of the nucleic acid, which is DNA and RNA, which has been surrounded by the protein codes known as capsule, which are used for protection of this virus against the host enzyme. So they are more smaller than living cell. So these are very small than living cell, meaning that they cannot be seen by using our naked eyes. They can only be seen using the scientific microscope, which is light microscope, where scientists mostly use them so as to magnify and can observe them. And the virus is not a cell. We say that the cell is a unit of life, and all living things are made up of cells. So from this scenario, meaning that virus is not a living thing. Now, the virology is the study of virus. So the study of virus is known as virology, and someone who is studying virus is known as virologist. Now, let us consider the characteristics of virus. We've already said that the virus lying in the boundary between the living and unliving things. Now we will see what are those characteristics which make the virus to become a living things, and what are the characteristics which make things become non-living. The first one, they do not have nuclear cytoplasm or organelles or cell organelles, meaning that the virus are non-living because they lack the component of the cell. That's why they do not have a cell. Also, they have a simple structure consisting of small piece of nucleic acid. So that's why we see they are small fragments of those nucleic acid which are surrounded by the protein coat, which is capsule. Also, some viruses have viral envelope enclosing the capsid, meaning that this viral envelope it functions as a membrane so as to maintain the shape of that particular virus. So just for the protection. Also, they cannot reproduce on their own. Virus they are not capable of reproducing by their own when they are outside cell, meaning that hawana uwezo wa kuweza kuzaliana wakiwa wenyewe, na hii sifa inafanya kuwa non-living things, meaning that the living things have the characteristics of reproduction. So this virus is not reproductive. They are not capable of reproducing when they are outside the host cell, even afanya kuwa non-living things. Na hivyo wanakuwa na uwezo wa kureproduce only when they are inside the host cell. Na hivyo wanaenda kutumia the nutrients from the host cell so as to reproduce. Now wanatumia obligate parasitism, meaning that this obligate parasitism, this is the form of relationship whereby the parasite can depend on the host so as to complete its life cycle, meaning that it cannot complete this life cycle without the host, or it cannot live without the host. Also, they have ability to crystallize outside the host cell. Kwa hivyo, wakiwa outside the host cell, virus wakiwa nje ya mwili wa any living things their host, meaning that wanaweza kuweza crystallize na kuwa kama. So that's why we are advised not to share this sharp equipment. In case if a person who have been infected by particular infectious virus diseases ambaye ametumia kile kitu, the same way pia kwenda kutumia kile kitu cha ncha kali, in case kama alijikata au alitumia au jasho na wewe kaenda ukakitumia kile kitu, in case kama that virus is transmitted either by blood or by using the sweet, meaning that wewe pia utakuwa infected. Kwa nini? Kwa sababu virus wana tabia za kuwa crystallizing outside the host cell. Na tumesema kwamba they are very small than the cell, meaning that hauwezi ukaona kwa macho yako. Kwa hivyo they tend to penetrate in body na kuweza kupelekea infectious diseases. Also, now let us see kwa namna gani sasa hawa virus wanaweza kaingia katika mwili wa host cell. Let's take an example. Host cell ni sisi binadamu. Human being is an example of the host cell for the human immunodeficiency virus, HIV virus. So human being is one of the example of the host cell for that particular HIV virus, ambapo anaenda yeye kazi yake ni kuweza kuvamia the white blood cell in a specialized cell inaitwa T helper cell. Now, let's take an example or the mechanism on how the virus can attack the host cell. The first one, the virus attach itself to the host cell and inject the DNA into the cell. Kwa hiyo virus kwanza anajiattach katika host cell. Take an example in the white blood cell. Baada ya kuattach pa then ana inject DNA zake. Baada ya injection, so hapa tunaangalia injection tayari hapa DNA zilikuwepo tayari zimebe injected hapa. The second process, that after the injection of this DNA, then undergo replication, meaning that they divide. They undergo genetic replication several times kuweza kutengeneza more DNA of this virus inside the host cell, ambapo inaweza kupelekea infection. Baada ya kureplicate, kitu gani kinakuwa kinafuata? After the replication, meaning that they will assemble na kuweza kutengeneza whole virus. So those genetic material inaenda kuassemble kuweza kutengeneza virus kizima. As we see from the first one, alikuwa ni whole virus ambayo alikuwa na attach hapa na kuweza kuinject this DNA. So after the replication, this DNA will assemble to form the whole virus. So the components assemble to form new virus which are complete with the protein code. So the protein code function is to provide the protection against the host enzyme. And the last one, the host cell bursts to release new virus. Baada ya assembling of the virus, now itaburst kwa ajili ya kuweza kurelease our hapa virus kwa ajili ya further infection. Na this virus when they will be released outside the host cell, meaning watakapokuwa wametolewa watakuwa non-living, meaning that they will crystallize in case mpaka pale ambapo tena watakapo invade another host cell. So when outside the host cell they show no symptom of life, so meaning that wanapokuwa outside the host cell wanakuwa non-living. Kwa hivyo undergo any type of cellular activity, kama vile hawezi kukua, do not grow, hawezi kufeed, they cannot excrete or respire, so they exist in a dominant state. Also, they are host specific, meaning that a virus is invading a particular host. Kwa virus, kwa mfano virus wa HIV ambaye kazi yake ni kuweza kuinvade the white blood cell, hawezi kwenda kuinvade the red blood cell. They are specific to their host. Virus ambao ni specific kwa ajili ya cow, hawezi akainvade binadamu. So meaning that they are host specific. They affect only specific host. So they are host specific. Certain virus attack specific host. Example, HIV attack only specific white blood cell in a specialized area known as T helper cell. Also, most viruses are infectious, meaning that wanasababisha magonjwa. Na hizo sababisha magonjwa ni kinatokea, they can end up of, they take an example of coronavirus. The virus which causes the coronavirus is known as COVID-19. So they are infectious, meaning that wanasababisha magonjwa ambayo ni corona, na hivyo ikapelekea millions of people wakafa kwa sababu ya ugonjwa. So they are very infectious. Now let us see the structure of virus. My lovely Form 1 students, virus have different structure depending on the type of the virus. Now let us consider their structure na shape zao. So viruses are composed of strand of genetic material, DNA or RNA, which form the core. Kwa katika at the center of the virus kuna genetic material ambayo inaweza ikawa either DNA or RNA. So the core is enclosed in a protein called capsid, as in bacteriophage. Kwa mfano katika bacteriophage, one of the example which of bacteria ambaye anakuwa ana affect other other bacteria, virus ambayo ana infect or invade other bacteria, anakuwa ana this protein coat ambayo inaitwa capsid kwa ajili ya protection against this host enzyme, ambazo hapa capsid kazi yake ni kuweza ku enclose or to surround this core which contain those genetic material. The DNA or RNA of various shape and size according to the types of organism. Kwa hiyo shape na size hizi RNA ni za tofauti tofauti kulingana na aina ya virus. Now let us see the shape and the structure of different virus depending on their structures. So we're starting with the first one here. This is the structure of the human immunodeficiency virus or shortly ni HIV virus, ambayo yenyewe amespecialize kumuaffect human being in the white blood cell. So this is structure tunaangalia within the code cytochrome which contain the DNA and RNA, tipo katika disk shape ambapo imebe surrounded with the protein coat ambayo inaitwa capsid. And this one is an envelope, ambayo envelope kazi yake ni kuweza to protect or to surround the inner contents of the cell. And inside hapa hizi hapa za ukijani hizi hapa nucleic acid na mimi naweza ikawa either any RNA or DNA in the central core. And then this one is a structure of the coronavirus. Oh, yes, this is the example of the coronavirus or COVID-19. Na this naangalia protein coat yake ambapo hivi hapa kuna nucleic acid yake ipo katika ring form, like spiral shaped. Hapa ni structure ya DNA. Kuna protein coat capsid kwa ajili ya protection against the host enzymes. Kuna envelope kwa ajili ya maintenance of the shape. Also ya the spike protein kwa ajili ya protein synthesis when they are for feeding inside the host cell. And this is an example of the bacterial phage ambayo kazi yake yeye mara nyingi anakuwa ana invade other bacteria. So and this is the capsid we surround the nucleic acid, and this is the collar. Also these are tail sheath as well as tail fiber kwa ajili ya attachment to the bacteria. Now, let us see the advantages of bacteria. My lovely Form 1 students, bacteria, although they are very infectious, they are very pathogenic. Also, they have numerous of advantages in different fields of biology. First one, they are important in the study of cellular and molecular biology to manipulate and investigate the function of the cell. Vile vile, hapa virus ni muhimu sana katika kwa wale watu ambao wa biologists ambao na specialize, kwa mfano microbiologist wanasoma microbiology, au hapa watu wa cell, watu wa genetics, zinasaidia kuweza kujua kazi the all different part of the cell they do function. Inasaidia kuweza kujua the life cycle of that particular virus, the how can be transmitted, na hivyo kuweza kujua dawa ambayo inaweza be designed so as to solve a particular or to cure a particular disease caused by the virus. Also, some viruses are used to make vaccine. Vile vile kuna baadhi ya virusi wanatumia kutengeneza chanjo. Chanjo, this is a substance that contains the weakened virus which is being injected into the body or can be in form of pill so as to be introduced into the body ya kuweza kureact so as to produce the antibody against the incoming pathogen. Kwa hiyo unapokuwa unapewa chanjo ni kwamba una be injected substance ambayo tayari wana vile virusi ili mwili wako uweze kujiandaa na kuweza kutengeneza antibodies against any incoming pathogen. Also, bacteriophage are virus that attack bacteria. Bacteriophage ni virus ambao wanattack bacteria. Kwa kama kuna wanakuwa wanattack bacteria, meaning that wanakuwa wana reduce those harmful bacteria from the surrounding. So this helps to control bacterial infection and diseases. Also, some viruses are used in biological control to eradicate pests such as insects. Vile vile kuna baadhi ya virusi ambao wanatumia katika biological control kwa ajili ya kuweza kuondoa some pest causing diseases kwa some plant crops ambazo za kupelekea the reduction of the yield in the farm. So these are one of the advantage of this virus wanasaidia katika kuweza ku reduce this pest causing insecticide in the farm. Now let us see the disadvantage of virus. Despite the virus have advantage in different fields of the biology in our daily life like production of the vaccine, also they have different disadvantage in our life. The first one, they are very infectious. My lovely Form 1 students, viruses are very infection as they cause diseases which affect and kill numerous or millions of people in our society, in our country, and in our world. Take an example for the coronavirus, which have been occurred very recently, which killed millions of people in the world. So, most viruses are pathogenic. They cause diseases and infections like polio, chickenpox. Kwa mfano kama polio, kuna tetekiwanga, na rabies ni kichaa cha mbwa. Kuna AIDS, kuna COVID-19. So those are the pathogenic diseases which have been caused by the bacteria. Also, pathogenic virus can reproduce very fast, leads to large scale epidemic. So hapa bacteria, pathogenic bacteria wana tabia kuweza kuzaliana kwa haraka. Na hivyo kwa mfano kama COVID, wanazaliana kwa haraka sana, hivyo kuweza kupelekea epidemic disease ambayo inaweza ka widespread worldwide within a short period of time. Also, virus can alter themselves often and thus become difficult to cure viral infection. Kwa wanaweza ka develop those resistant gene, or they can mutate, hivyo wakawa resistant to any drug which has been designed for us to cure them. Kwa inakuwa ni vigumu sana kuweza kutiwa ugonjwa, hivyo unaweza kupelekea mtu kufariki. For example, there are many different types of virus that can cause common cold and influenza. Na hivyo inapelekea kuna madawa mengi sana yenye kutibu mafua pamoja na influenza. Now, my lovely students, now I hope that you have enjoyed the lesson and now you are familiar with the types of the virus, characteristics of virus, the advantage as well as disadvantage. Before I sum up my session, you have to take a note on this test so as to improve your understanding and make other follow-up. The first question states that, explain why viruses are considered as both living and non-living things. Explain why viruses are considered as living and both non-living things. The second question, describe the structures of bacteriophage. You have to describe the structure of bacteriophage. The third question, explain the economic importance of virus. When you're told to describe the economic importance, here you will mention both positive and negative, or advantage and disadvantage of the bacteria. Up to now, my lovely students, have nothing to say. Have a nice moment.